Welcome back to the Deep Dive. I'm your host, and today, wow, we are venturing into a really fascinating area, sort of where uh, fundamental biology meets the very practical world of physical recovery.
Yeah, it's a cool intersection.
Totally. We're diving into the story of two peptides, BPC-157 and TB-500. And these are compounds that have definitely, captured attention, sparked a lot of research,
Oh, absolutely.
and generated, well, a fair bit of buzz, right? Whispers in regenerative medicine circles, recovery
You
communities.
hear the names a lot. BPC, TB.
Exactly. And with me, as always, is our expert, a clinician researcher, who really digs into this regenerative science stuff. I'm genuinely looking forward to unpacking this one. It feels like it could shed light on some really cutting-edge ideas in healing.
It's an excellent topic. Yeah, I share your enthusiasm. It's always rewarding to explore these areas where our understanding of the body's own healing abilities, how it works, starts to point towards potential new strategies for repair, regeneration. And BPC-157 and TB-500, especially when you think about them together, they really offer a compelling look at that frontier.
Okay, so for you listening in on our deep dive today, here's the plan. We're going to unpack these two compounds, BPC-157 and TB-500, systematically.
Right.
First, where did they even come from? Their origins are pretty different, apparent different.
Yeah.
Then we'll dig into why researchers and, people in recovery fields often talk about them together, this whole idea of potential synergy.
The combination aspect. Crucial.
Yeah. And we'll get into the mechanisms. How did they supposedly work? Like pull back the curtain on the biology.
The nitty-gritty.
Exactly. Then look at the research, mostly preclinical, I gather, especially for soft tissue stuff.
Correct. Lots of preclinical work.
And finally, kind of gaze into the crystal ball a bit. What's the future look like for this duo in science? But always, always with the caveats, right? Because this is evolving science.
Definitely not settled science. Important to remember. It's a journey, really, from finding these molecules to thinking about, real-world impact. Lots of fascinating details. So let's start at the start.
Let's do it. All right, origins. Let's unpack BPC-157 and TB-500. Where did they actually come from? Because like you said, their origins are really distinct. It's like they started on totally separate scientific paths.
They absolutely did, yeah. Let's start with BPC-157. People often call it body protection compound 157.
Right.
This one has a really unique origin story. It was actually identified from a protected protein that's naturally found in our own gastric juice.
Gastric juice, stomach acid environment.
Exactly. Think about it. The stomach, it's one of the harshest places in the body. Super strong acids, enzymes.
Yeah, it's amazing it doesn't just dissolve itself.
Right. But it has this incredible ability to protect its own lining, to heal itself. And this little peptide, just 15 amino acids long, was identified back in the 1990s, connected to human gastric research. It's part of the stomach's own defense system.
That is genuinely wild. So our stomach has its own kind of molecular bodyguard, and scientists basically found it. What did they do first once they isolated it?
Well, the initial discovery was really driven by watching how the stomach protects itself from, injury. So the early preclinical work, mostly in animal models like rats, showed pretty remarkably that BPC-157 could prevent stomach ulcers and even actively heal them. Ulcers caused by all sorts of things, alcohol, NSAIDs, stress.
Wow.
Yeah, it was a really strong, clear signal, a natural repair mechanism just doing its thing, suggesting it wasn't just protective, but actually regenerative right there in the gut lining.
Okay, so it starts in the gut.
Yeah.
Which, you don't immediately think torn muscle or sprained ankle when you think stomach lining.
Not at all.
But then, science being science, curiosity kicked in, right? They must have thought, if it heals the stomach so well, what else can it do?
Precisely.
Yeah.
That's exactly what happened. Those initial gastric findings were just so compelling, it sparked broader questions. The same research groups, and then others later on, started looking beyond the GI tract.
Right.
And what they reported in rodent studies after that was, pretty remarkable. Signals suggesting it could help repair a whole range of different tissues.
Like what?
Muscle, tendon, ligament, even nerve tissue, brain tissue. After different kinds of experimental injuries. This really broad spectrum of potential repair activity is what lit the fuse, Got everyone interested in BPC-157 as a research tool.
So it's like this peptide, born in the tough environment of the gut, somehow learned a kind of universal repair language the body understands elsewhere.
It's a great way to put it, yeah. Like it developed a more general repair instruction set. It's even been looked at in some early clinical settings, things like inflammatory bowel disease. And the reports mentioned encouraging signals and a favorable short-term safety profile, at least in the animal models they were using. But, and this is super important, we'll keep saying this, it is not an approved drug.
Right. Still experimental.
Absolutely. It lacks those big, large-scale, placebo-controlled human studies you need for regulatory approval for actual clinical use. It's still firmly in the research compound category.
Got it. So BPC-157, the quiet hero from the stomach's defense force, surprising everyone with its broad repair signals and preclinical work. Okay, let's pivot. TB-500, how does that fit in? Its journey was different, I understand.
Completely different path, yeah. TB-500 wasn't directly isolated from nature in its final form like BPC-157 was. It's actually a lab-made fragment.
Synthetic.
Right, a smaller synthetic piece designed from a much larger protein that is naturally occurring. That one's called thymosin beta-4, or T4. It has 43 amino acids.
I'm a beta-4.
And what does the natural T-beta-4 do? Oh, T4 is fascinating in itself. It's found pretty much everywhere in the body, almost ubiquitous, and it's involved in tons of crucial biological processes. Think of it like a master orchestrator for fundamental stuff cells do, like moving around cell migration, growing new blood vessels, that's angiogenesis, and just general tissue remodeling after an injury. It's really foundational for how our bodies respond to damage and keep tissues healthy.
So if T4 is natural and so important, why make TB-500? Why not just use the original P4 protein? What was the point of engineering this smaller fragment?
Excellent question. Very practical. The full T4 molecule, even though it's potent, it's pretty large, biologically speaking, and in its natural state, enzymes in the body break it down quite quickly.
So it doesn't stick around long.
Exactly. That limits its stability, its effective half-life, and that means it limits how widely and effectively it can spread through the body to do its job. So researchers, mostly in the 2000s, maybe into the 2010s, they engineered TB-500 specifically to capture the key biologically active parts of TOFOR, its sort of core functions, but at the same time, improve its stability and its ability to reach tissues systemically in experiments.
So make it more stable, last longer, travel better.
Precisely. The goal was a better research tool, more stable, more bioavailable, to study TOFOR's regenerative potential more effectively. And just like BPC-157, super important to remember, TB-500 is not approved for medical use.
Right. Research only.
Typically available, where it's lawful, for research purposes only. Strict guidelines.
Okay, so summing up the origins. BPC-157, roots in the stomach's protection system, then showed broader repair signals. TB-500, a clever synthetic fragment inspired by this widespread natural orchestrator protein, T4, designed to help cells move and rebuild more effectively systemically.
Exactly. You nailed it. Different sources, different initial discoveries, one from the gut's own defense system, the other engineered from a protein that helps cells move and rebuild. But they both end up converging on this really exciting area, repair biology. They emerged as research tools because of all those promising preclinical signals, not because they were developed as commercial drugs from the get-go.
Which leads perfectly to the next big question. Why? Why, despite these very different origins, are BPC-157 and TB-500 so often discussed together? What's the thinking behind pairing them up in research or even just in conversation?
Yeah, this is where it gets really interesting. The core reason they're often looked at together, the hypothesis driving it, really boils down to one word: complementarity.
Complementarity, okay.
The idea is that together they might cover more bases, more phases, more aspects of the whole complex repair process than either one could manage on its own. It's not like having two players doing the exact same job. It's more like having a specialized team where each member brings unique strengths to solve a multifaceted problem.
Okay, so if we go back to our construction site analogy, which I find super helpful for this stuff, BPC-157 is often seen as the one doing the local work, right? Like prepping the specific site of the injury.
You're exactly on the right track. BPC-157's preclinical signals often suggest it acts like the local site preparer. It optimizes the immediate microenvironment right at the injury, makes sure the conditions are, well, perfect for healing to start.
Okay.
In contrast, TB-500 is thought to be more systemic in its influence. It's linked to broader effects, like boosting cell migration, literally helping the workers get where they need to go, and coordinating the bigger picture of tissue remodeling throughout the body.
So less about the specific patch of ground, more about the overall logistics.
Precisely. It's about the efficient flow of resources and personnel across the whole project.
That's a great way to visualize it. So BPC-157 is down there, prepping the site, laying the foundations, getting the local supply lines running smoothly,
Mm.
while TB-500 is kind of coordinating from above, making sure the workers get there,
Mm.
organizing the whole rebuilding effort system-wide. It really does sound synergistic.
It perfectly captures the working model, yeah. The hypothesis that many researchers and folks in recovery communities talk about. The hope is that by addressing both the immediate local needs and the broader systemic coordination,
Right.
you might get a more robust, more efficient, maybe even higher quality healing outcome, especially for those injuries that just don't heal well on their own.
Okay, beyond the construction analogy, what are the more specific sort of biological arguments for combining them? What theoretical advantages are commonly cited?
Well, a big one is that they seem to target different but complementary bottlenecks in that whole healing cascade.
Bottlenecks, okay.
Yeah. So BPC-157, for example, is often linked to promoting local angiogenesis, getting new blood vessels right where they're needed, and helping organize the collagen, the matrix, the structure.
Building the scaffold.
Exactly. TB-500, drawing from its 2,4 heritage, is more connected to enhancing cell migration systemically, supporting the health of blood vessel linings generally, and really guiding the quality of the remodeling process, aiming for less scar tissue.
Less scarring, that's key.
Absolutely. So the theory is they're not just doing the same thing, they're tackling distinct, vital parts of the job. One preps the ground, the other orchestrates the build.
And that local versus systemic difference you mentioned earlier, how does that play into the theoretical advantage?
Right. BPC-157 has been studied a lot for local use injected right at the injury site in preclinical models, or even taken orally for gut issues, acting directly. TB-500, designed for systemic spread, can theoretically exert its influence more broadly, mobilizing cells, supporting blood vessels everywhere. So the pairing aims for full coverage, hitting the specific microdynamics at the injury and supporting the macrodynamics of the whole system. Comprehensive support.
Now, I have definitely heard, not just in research papers, but, online, in fitness circles, talk about anecdotal momentum with this pair. What's that about?
It's definitely out there, yeah. In recovery communities, you hear it from some coaches, some physiotherapists who are looking at cutting-edge stuff. There are quite a few anecdotal reports, often describing, quicker turnarounds for really stubborn injuries, soft tissue stuff that just wouldn't heal. When both BPC-157 and TB-500 were used, in research contexts, obviously, and this kind of grassroots interest, while it's not formal proof, it definitely creates buzz, fuels the conversation, hints at something potentially effective that deserves more rigorous study.
And is this where that nickname comes from, the Wolverine stack, reflecting that hope for super fast healing?
Yes, that's exactly where it comes from. That informal name definitely captures the enthusiasm, the mystique around these peptides.
Yeah.
You see it in forums, athlete discussions, even some clinicians exploring new things. It's shorthand for that hope of accelerated high-quality recovery, almost like the comic book character, right?
Right.
But it's crucial to remember it is just a nickname. It reflects that desire for rapid, maybe even fictional level healing.
Yeah.
It speaks more to the need for better therapies for tough injuries rather than verified outcomes.
Good point. It's enthusiasm, not proof.
Exactly. Take it figuratively. It highlights a real gap where people are searching for answers, but it's not established science.
Which brings us right back to the critical caveat.
Yeah.
This whole idea of combined use, while it sounds great theoretically and has these anecdotal reports, it's still mostly a hypothesis, right? Not proven fact.
Absolutely. Crucial caveat. The theory is compelling, the preclinical data is growing, the anecdotes are intriguing, they all push research forward. But definitive proof that the combination is truly better than either one alone or better than standard care.
Yeah.
That requires those rigorous, gold standard, controlled head-to-head trials in humans. And we're just not there yet with that level of solid clinical data.
Okay.
It's a really active, exciting area of research interest for sure. But validating true synergy, that's still very much a work in progress, an emerging frontier.
Okay, let's get into the weeds a bit, the mechanisms. How do these peptides actually work at the cellular level? Let's bring back our construction analogy. For a good repair job, you need supplies, workers, coordination, and cleanup, right?
Perfect framework, yeah. Let's start with BPC-157, our local site preparer and protector. Its actions are diverse, but they really focus on optimizing that immediate area right at the injury.
Preparing the ground, laying the pipes. How does it do that biologically?
Okay, one of the most well-documented things in preclinical models is its link to angiogenesis.
New blood vessels.
Exactly, forming new microvessels, like opening up essential capillary side streets. BPC-157 seems to boost pro-angiogenic signals, things involving VEGF pathways.
VEGF, vascular endothelial growth factor.
Right, critical for vessel formation. It also seems to affect nitric oxide pathways, helping relax blood vessels. So, the practical result: better local blood flow,
More perfusion.
significantly improved local perfusion, which means much better delivery of oxygen, nutrients, immune cells, right to those tissues that often heal slowly because they don't have great blood supply, like tendons. It's laying down those vital supply lines.
Makes sense. Got to get the materials there. What other local prep work is it doing?
It also plays a big role in matrix and collagen organization.
The structure.
Yeah, the structural stuff. Research points to things like enhanced fibroblast activity. Those are the cells that make collagen, the protein that gives tissue strength.
Mm-hmm.
It also seems to promote tenocyte outgrowth in tendon models. Tenocytes are the specific cells for tendon repair. And crucially, it seems to lead to more orderly, functional collagen, not just scar tissue.
So better quality building blocks, laid down properly, not just a messy patch.
That's the idea. Stronger, more organized, more functional tissue, closer to the original.
Yeah.
And beyond structure, BPC-157 is often described as cytoprotective.
Cell protecting.
Exactly, helping protect cells from damage and stress during injury. Plus, it seems to shift the inflammation away from being prolonged and damaging
Right.
towards a more constructive, repair-focused state, like keeping the neighborhood calm enough to rebuild, making sure inflammation does its initial job but doesn't become the problem.
Okay, that makes a lot of sense. And you mentioned nerve and barrier effects briefly earlier.
Yeah, some exploratory preclinical work has shown neuroprotective signals, helping protect nerve cells, maybe aiding nerve regeneration. Very interesting area.
Wow.
And tying back to its origins, it shows really robust support for the GI mucosal barrier, helping keep the gut lining intact. So, summing up BPC-157, it's often seen as optimizing the local site, improving blood flow, supporting matrix structure, protecting cells, balancing inflammation locally, a powerful local organizer.
Really multifaceted. Okay, now let's switch to TB-500, the systemic mobilizer and organizer.
Mhm.
How does it contribute, and how is its mechanism different?
TB-500's role, heavily influenced by its parent, P4, is more about getting the workers to the site and organizing them effectively. A key mechanism seems to be its ability to bind G-actin and influence the cytoskeleton.
Okay, actin, cytoskeleton, simplify that.
Right, so actin is basically the internal scaffolding in cells. It lets them keep their shape, divide, and really importantly here, move.
Movement.
Yes. By tweaking this internal scaffolding, TB-500 seems to facilitate efficient cell movement, getting key cells like fibroblasts, endothelial cells for blood vessels, immune cells, getting them to migrate towards the injury site, helping the workers actually get there, like an efficient transport system.
So it's managing workforce logistics. Totally different job, but just as vital.
Yeah.
Does it also help with angiogenesis like BPC-157?
It does, yeah. And it's a great example of that complementarity. While BPC-157 might be opening local side streets, TB-500 is associated with pro-angiogenic signaling and endothelial support more system-wide.
Broader effect.
Right, helping microcirculation, supporting adaptive blood flow control throughout the whole body. So if BPC ensures good local supply, TB-500 helps ensure the main vascular highways are working well, feeding all the repair sites.
Okay, and it has a role in remodeling too, beyond just patching things up.
A really important one. A key thing highlighted in the T4 literature is promoting organized regeneration with less dense, restrictive scarring. Fibrosis-aware remodeling, they call it.
Fibrosis aware, I like that.
Yeah, because you don't just want a wound closed, you want the tissue to get back its flexibility, its function. TB-500 seems to help guide remodeling towards that functional outcome, reducing that excessive tight scarring that can limit movement and cause pain later.
Like ensuring the rebuilt wall isn't just solid, but also flexible like the original.
Perfect analogy, because yeah, what good is healing if you're left with a stiff, limiting scar?
That's a huge point. Think about athletes dealing with lingering limitations from scar tissue, not just the injury itself.
Yeah.
Okay, so summarizing TB-500, logistics coordinator, gets cells where they need to go,
Yeah.
supports vasculature broadly, guides remodeling towards function.
Exactly. So when you think about them working together, that complementary story gets really clear. BPC-157 seems to focus on site prep, local blood flow, matrix organization, cell protection. TB-500 focuses on system logistics, cell migration, broad endothelial support, and that crucial fibrosis-aware remodeling.
So one cultivates the perfect local environment, the other mobilizes and organizes the workforce and materials. Sounds like a dream team, theoretically.
That's the model researchers are exploring, yeah. It's biologically elegant, but always have to keep the considerations in mind.
Right.
Because angiogenesis, growth, signaling, these are powerful processes. Researchers are rightly attentive to theoretical concerns. Could these pro-growth signals be wrong in certain situations? We need more study on long-term safety. And
Absolutely.
of course, individuals always respond differently. Biology isn't one size fits all.
Okay, let's shift to specific research areas.
Yeah.
Tendons, ligaments, soft tissue. These are often the really tough injuries, right? The heartbreak injuries that heal slowly and frustrate everyone.
Absolutely, the heartbreak injuries, yeah. Tendons, ligaments, muscle, fascia. Huge challenge in sports medicine, orthopedics, recovery in general.
Why are they so tough to heal?
It's usually a mix of things. Often they just don't have a great natural blood supply.
Less resources getting in.
Exactly. And they're under constant mechanical stress from just moving around, which can disrupt healing. So you get this slow, often incomplete healing, chronic pain, high risk of re-injury. Incredibly frustrating.
Yeah, the ones that just linger.
They really test your patience, often don't fully recover without significant help.
So given those challenges, what has the preclinical research shown specifically for BPC-157 in these kinds of injuries? Tendons, ligaments?
The preclinical work for BPC-157 here has been pretty consistent and, well, compelling. Lots of reports. Studies in animal models like rat Achilles tendons, patellar tendon models.
Okay.
They've reported some really interesting findings. Things like enhanced tenocyte outgrowth, getting more tendon-building cells into the area, improved cell survival and migration, making sure those cells get there and stay viable, better bridging of the tear itself.
Closing the gap.
Right, and forming earlier, more orderly collagen. And crucially, this better collagen structure was linked to stronger mechanical properties in the repaired tissue.
So not just patched, but stronger, more functional.
Closer to the original healthy tissue, yeah.
Mm.
And similar results, faster healing, better structure reported in ligament models, muscle injury models too. Always these themes of better blood flow, faster organization, even some preclinical signals for bone healing.
Wow. That's impressive for something first found in stomach juice. Speaks to that universal repair language, maybe. Okay, how about TB-500 and these connective tissues? What does the research show there?
TB-500, drawing from that big T4 literature base, shows different but equally important signals. Consistently linked with improved vascularity, better blood vessel supply to the area, vital. Also enhances cell migration significantly, getting the repair crews mobilized. And a really standout finding is its role in reducing adhesions and fibrosis, scar tissue. That remodeling quality signal comes up again and again.
Less restrictive scarring.
Exactly. Helping tissues heal functionally, maintaining flexibility and range of motion. The broader T4 literature also mentions things like skin ulcer healing, corneal healing, showing its role in quality repair in other tissues too. Even signals for cardiac, muscle regeneration in animals for the TEF4 family suggest a really widespread role in tissue repair systemically.
So quality repair is a big theme for TB-500. I think you mentioned potential beyond just tendons and ligaments, broader soft tissues, joints.
Yeah, exactly. Beyond direct repair of a tear, there's early preclinical work and some, small reports from the research community suggesting a role in modulating inflammation within joints like synovitis and contributing to more orderly repair alongside standard stuff like physio.
Right.
For example, some clinics, some researchers have explored pairing these peptides with things like PRP, platelet-rich plasma, hypothesizing synergy.
Less caveat time.
Big caveat time. Vital to stress. Formal, well-designed clinical trials are absolutely needed to actually quantify any added value, confirm synergy, establish proper protocols for combinations like that.
Which brings us back to that crucial distinction.
Yeah.
Preclinical versus clinical.
Indeed. Cannot stress this enough. Most data remain preclinical. The bulk of this exciting evidence, animal models, lab dish studies. And while that's super encouraging, translating that reliably to humans is complex. It takes time, needs those rigorous, controlled human trials, looking at timing, dosing, different people, long-term safety, real-world outcomes.
Right.
So we're talking encouraging signals, potent biology, but not guarantees, not approved treatments. It's cautious optimism driving the research forward.
Let's dive back into that synergy idea. That phrase people use, 1 + 1 is greater than 2. Where does that potential power come from when we talk BPC-157 and TB-500 together?
Mechanistically, yeah, the synergy idea is quite plausible, biologically speaking. Like we said, BPC-157 seems great at enhancing local tissue receptivity, better blood flow, friendlier environment, prepping the site,
Mm-hmm.
while TB-500 helps mobilize and organize the workforce systemically, getting cells there, coordinating the rebuild. The synergy potential comes because these actions aren't the same. They're distinct but deeply interconnected.
So not redundant. They're tackling different parts of the same problem, like gears meshing together for a bigger effect.
Exactly. That's a perfect way to think about it. It could mean, for instance, if BPC 157 is uptuning how local tissues respond, maybe making them more sensitive to growth factors via VEGF pathways, optimizing nitric oxide locally, making the injury site super receptive.
Okay.
Then TB-500 comes in, promoting factor release and cell migration system-wide. They could be mutually reinforcing. BPC sets the stage perfectly. TB ensures the full cast and crew arrive and know exactly what to do. It's a plausible model, but definitely needs direct testing.
It sounds like they could be hitting multiple bottlenecks in the healing process, one after the other, just streamlining the whole thing.
That's the core hypothesis, yeah.
Yeah.
By combining agents that target different bottlenecks across the healing phases, inflammation, proliferation, remodeling, they might collectively speed things up or improve the final quality.
Right.
Which could explain faster subjective recovery times people report anecdotally, or maybe lead to more complete functional repair than either could do alone, optimizing the whole continuum.
And we keep coming back to those anecdotes. While we know they spark interest, we have to keep their limitations in mind, especially for synergy claims.
Yes, absolutely. Treat them with scientific caution. Like we said, some coaches, therapists, individuals do report quicker results for stubborn injuries when both are used in research settings. Valuable for hypotheses, but
Not controlled trials.
definitely not controlled trials. Definitive synergy, scientifically. Needs those gold standard head-to-head comparisons, combo versus single agents versus placebo. In robust human studies, the clean proof just isn't written yet. Needs rigorous, peer-reviewed research.
And combining any active biological agents introduces more complexity, right? More things to consider.
Absolutely critical point. Combining agents can compound uncertainty, optimal timing, dosing for the combination, potential interactions between the peptides themselves, or with other things someone might be taking.
Yeah.
That's why any exploration needs careful supervision in ethical research settings, and why quality, purity, verification of the materials are non-negotiable. Biological systems are complex. We have to be meticulous.
Okay, let's switch gears slightly. If someone's just curious about how BPC-157 and TB-500 stack up individually, what are the key differences? They both target repair, but they have distinct personalities, it seems.
They really do. It's a great question. In terms of origin and structure, BPC-157 is that smaller 15 amino acid peptide tied to gastric research historically, known for that unusual oral stability in gut studies.
Right, can survive the stomach.
Yeah, though it's also used via injection in research for other targets. TB-500, on the other hand, is that synthetic fragment based on the bigger 43 amino acid TEF4, engineered for systemic bioavailability, usually given by injections subcutaneous, intramuscular in research, aiming for wide distribution.
So BPC has that unique oral angle for gut stuff. What about their main strengths or use cases when discussed individually in the research world?
Research discussions often frame BPC-157's strengths around localized soft tissue support, tendons, ligaments, muscles, often injected locally in models, and of course that strong GI barrier support linked to its origins, plus those exploratory neuroprotective signals, so often seen as more site-focused except for the GI work.
Okay, and TB-500?
TB-500 is known more for its systemic distribution. The emphasis is on facilitating widespread cell migration, supporting microcirculation, and that fibrosis-aware remodeling. It's been looked at across a wider variety of tissues, skin, eyes, in the T4 literature, pointing to a more global, systemic influence on healing.
So BPC for targeted local issues, TB for broader systemic effects. Is that a fair shorthand?
That's generally how researchers frame their primary strengths, yeah. And that difference shows up in their effect profiles, like angiogenesis, new blood vessels. Both promote it, but BPC is often seen as very local site-oriented, boosting perfusion right there. TB-500 is framed as more system-wide support for vascular health, the bigger network.
What about inflammation? How do they compare individually there?
Also different emphasis. Both seem to help balance inflammation. But TB-500 via TFO4 is often discussed for broader cytokine modulation, regulating the overall inflammatory response systemically. BPC-157's signal is often described as optimizing the local niche, keeping inflammation constructive at the site.
Okay. Any other key differences? Tissue specificity, administration?
Yeah, BPC has that very distinct GI barrier focus preclinically, logical link to its origin. The TB-500 TIF4 literature covers more ground skin, cornea, alongside soft tissues, suggests a more universal cellular role. Administration-wise, BPC explored orally, GI, and locally via injection. TB-500 typically via injection for systemic effects in research.
So when researchers or others are thinking which one for what situation, what's the common practical framing they might use to distinguish them?
The common practical framing you sometimes hear in discussions, maybe informal protocols, is that for really localized specific targets like one particular tendon tear, the conversation might lean towards BPC-157 for its local prep strength. If the issue is more multi-site recovery, general soreness, systemic overuse, or needing broad cell mobilization, then TB-500 often comes up for systemic use. But honestly, many theoretical protocols, especially driven by anecdotes, quickly consider both for that complementary coverage, like having two different tools in the same toolbox, addressing different aspects of the same problem. But again, that's pending the actual head-to-head data.
Right. Always back to the need for data. And what about reported tolerability short-term in research contexts?
Generally, both are reported as pretty well tolerated short-term in preclinical and early studies. That's a positive signal.
Any side effects mentioned?
Anecdotally, some individuals mention transient things, headache, maybe light dizziness, mild fatigue, sometimes reported a bit more often with systemic TB-500. But, and this is critical, rigorous, long-term safety data in humans, that's still a major need. We really don't know enough about sustained use yet. Hashtag safety, boundaries, and the path forward.
Okay, this brings us to a super important part. Safety, the rules, the boundaries, and what needs to happen next with this research. What are the absolute must-know safety and regulatory points for anyone listening?
This might be the most critical takeaway, honestly. First, maybe bold and underlined, these are research compounds only in this context.
Not approved drugs.
Not approved drugs, not cleared as treatments by the FDA or similar bodies elsewhere. They are not for routine medical use. Period. That distinction is fundamental. They're investigational. Safety and efficacy in humans are still being studied, not established.
And the biology they tap into is powerful.
Right.
Which needs caution.
Exactly. The very pathways they influence angiogenesis, cell growth, are potent. Fundamental biology. That's why they're interesting, but it also demands incredibly thoughtful, careful evaluation in formal studies, especially for long-term use.
Right.
There are always theoretical risks with stimulating growth or blood vessel formation. Could there be off-target effects? Could it be wrong in certain contexts? These need thorough investigation in controlled settings.
And the practical side, getting the actual stuff. Quality matters hugely here.
Massively. Quality and sourcing are absolutely crucial. In any research, you need purity, accurate labeling, verification. You have to know you're working with the real deal, no contaminants.
Otherwise, the results mean nothing.
Exactly. It's foundational for reliable research. And frankly, it's a huge challenge in an unregulated market where these might be found.
So for anyone listening who's maybe dealing with an injury, thinking of a recovery, this stuff doesn't replace the basics, does it?
Absolutely not. Couldn't be clear. None of this replaces foundational rehab. Proper loading, rest, guided physical therapy, those are still the main drivers of good recovery outcomes.
Right.
These peptides are interesting tools being studied, maybe to optimize biology, but the fundamentals, physio, medical guidance, they still rule. And one more really important point, especially for athletes, TB-500 and related compounds are prohibited in professional sports.
Banned by WOTA.
Yes, by WOTA and similar organizations. Both peptides remain experimental, research only. Athletes, coaches must know these rules.
Okay, so given all that, where does the field go next? What are the big steps needed for this research to move forward, to get clearer answers?
Several crucial fronts need progress, really.
Like what?
1, rigorous clinical trials. Top priority. Move from animals and anecdotes to controlled human studies, comparing combo versus single agents versus placebo for specific injuries with standardized rehab objective measures. That's how we get real answers.
Okay, what else?
2, long-term safety profiling and toxicology. We need way more data on long-term safety, optimal dose ranges, how they affect different groups of people, and thoroughly investigating those theoretical risks around growth and angiogenesis. Understand the full picture.
Makes sense.
3, optimal dosing and delivery, figuring out the best timing during healing, evaluating oral BPC-157 properly, is it just for the gut, or does enough get systemic? Studying different injection routes, maybe even long-acting versions or targeted delivery, maximize effect, minimize
Like
risks.
deeper science, too.
4. Deeper mechanistic mapping, really understanding how they work at the receptor level. Actin dynamics, endothelial effects. Matrix remodeling. This helps find biomarkers, ways to predict who might respond best, moving towards personalized approaches.
Okay.
5. Broader therapeutic exploration. Hypothesis-driven studies in other conditions, inflammatory, fibrotic, degenerative, and carefully controlled studies pairing them with existing things like PT or PRP to prove if there's added benefit, not just assume it.
And finally.
6. Regulatory and pharmaceutical development. If efficacy and safety are strongly proven through all that work, then you'd see efforts towards standardized, quality-controlled pharmaceutical products with clear approved uses. That's the long-term path.
So the ultimate goal is basically replacing guesswork with solid data.
Exactly. Clarity. Where does it help? Who does it help? Under what exact evidence-based protocol? Less speculation, much more data. That's the scientific goal. Hashtag tag out true.
All right, quick recap then. BPC-157 and TB-500. Fascinatingly different origins, stomach protection versus engineered systemic orchestrator. But they might complement each other beautifully in healing,
Yeah, potential.
influencing local blood flow, cell movement, remodeling quality, really encouraging signals in preclinical work, especially for soft tissue. And that combo idea, compelling. But,
But
the big but.
we absolutely still need those definitive controlled human trials to prove synergy, establish safety, figure out if and how they fit into clinical practice. Balanced, curious, but cautious.
Perfectly summarized. That's the approach we need for this promising but still very much emerging field. It's a discovery process.
Excellent. Thank you so much for walking us through this. Always insightful, always a pleasure to get your perspective.
My pleasure. Always enjoy the deep dives. Before we wrap up, just a quick and very important note for everyone listening. The mentioned products and peptides are not intended to refer to any peptides or products sold by Power Peptides. Products sold are strictly for research purposes and not intended for human consumption. Thanks again for joining us for the deep dive. If you found this look into regenerative science as fascinating as we did, please hit follow or subscribe wherever you listen.
Yeah, stay tuned.
And if you know someone really curious about the future of recovery, maybe dealing with those stubborn injuries, share this deep dive with them.
Spread the knowledge.
So, as we keep exploring our body's amazing healing powers, here's a thought to leave you with. What advancements do you think are just around the corner, waiting to totally transform how we approach recovery and regeneration? Chew on that one. We'll see you next time.